It is often said that DNA is the instruction manual of life. This is true, for it is the genetic information contained in an organism's cells that determines how those cells operate, and therefore the form and functioning of the organism. But what happens when there are mistakes, or mutations, in the instruction manual of life? The short answer is that life doesn't go very well. In the best cases, the result can be a lifetime of pain, suffering, and disability. In the worst cases, it can be death. For 3.5 billion years, there was only one solution to this problem. Those organisms with intolerable mutations simply had to get along as best they could, most likely only to then die young and childless. The situation is different, however, for the human organism. We are the one species with the potential to take control of our genetic destiny. We can rewrite our own instruction manual. Just in the last decade, we have developed an entire suite of techniques for editing DNA, giving us an unprecedented mastery over the human genome. This is the CRISPR revolution. By the way, if my voice sounds slightly unnatural, you can rest assured that it is not because of genetic modification. I have a severe speech impediment due to having cerebral palsy. CP is not a genetic disease, but a movement disorder caused by early brain injury. However, one of the stated missions of this channel is to help push humanity towards a post-disability world, and this includes those whose disabilities are genetically caused. Luckily, my speech impediment is not an impediment to this mission, thanks to Murph. Murph uses state-of-the-art AI software to generate artificial voiceovers. I just type in what I want to say, and Murph says it clearly for me. If you think that you might want to use Murph for your own video projects, follow the link in the description. In this video, we're going to take a look at the CRISPR Toolkit. This is a set of molecular tools which allow us to go into living cells and make physical changes to their genomes, altering how they function. Clinical trials are already underway. People are being set free from painful and debilitating genetic diseases that were once incurable. And there is every reason to believe that this will continue. Thanks to the tools showcased here, and others like them yet to be developed, we may soon see a day when such conditions are a thing of the past. Part 1. CRISPR-Cas9. The molecular scissors. The first gene editing tool that we must examine is the CRISPR-Cas9 system itself. This is true not only because it began the revolution, but also because all of the other tools that we will discuss are essentially just elaborations of it. Its basis evolved as part of the adaptive immune system of bacteria. Bacterial cells are sometimes infected by a specialized type of virus known as a bacteriophage. As you may know, viruses work by sneaking their DNA into a cell and tricking its internal machinery into making more virus particles. Upon surviving such an attack, bacteria have a way of remembering the bacteriophage, so as to not be fooled by it again. They snip out a piece of the virus's DNA and incorporate it into their own. A bacterial cell may have several such viral snippets stored in its genome, likely from different bacteriophages. These are separated by short sequences, called clustered regularly interspaced palindromic repeats, from which the CRISPR-Cas9 system gets half of its name. They are palindromic because they are the same whether read forward or backward, like the words race car or kayak. When a viral snippet plus the palindromic repeat that immediately follows it are transcribed into RNA, they form what's called CRISPR RNA or cRNA. The other half of the CRISPR-Cas9 system is CRISPR-associated nuclease 9 or Cas9. A nuclease is a type of enzyme that induces double-stranded breaks, or DSBs, in DNA. To attach the cRNA to the Cas9 enzyme, a third component, called transactivating cRNA or tracer RNA, is needed. Like cRNA, tracer RNA has two parts: a nucleotide sequence that complexes with the Cas9, and another palindromic repeat. Because they are the same forward and backward, the two palindromic repeats anneal to each other. This combines the two vital components while leaving the viral snippet free. When the bacterium is reinfected, the Cas9 enzyme uses the now attached snippet to recognize the viral DNA. When it finds a match, the Cas9 unwinds the DNA and uses the viral snippet to line itself up, before precisely cutting both strands. The DNA is then glued back together by the cell's natural repair mechanisms, in a process called non-homologous end joining, during which nucleotides are randomly added or subtracted between the two endpoints. These alterations are referred to as indels, a portmanteau of insertion and deletion. The introduction of indels effectively scrambles the virus's genetic code enough that it can no longer be transcribed and is thus rendered inert. The CRISPR revolution was kicked off when scientists found that cRNA and tracer RNA could be combined by chemically linking the two palindromic repeats together to form a loop. The creation of this new type of RNA, called single-guide RNA or sgRNA, greatly simplified things. It could be synthesized in a lab and, combined with Cas9 enzymes harvested from the bacteria Streptococcus pyogenes, made to target any nucleotide sequence of about 20 base pairs long. The combined system cuts the DNA at an exact spot specified by the sgRNA, like a pair of molecular scissors. 4 types of DNA edits are possible with these scissors: disabling, deleting, inserting, and replacing. The simplest is the disabling of troublesome genes, which is done the same way as the virus's genome. To delete a gene, 2 Cas9 complexes are used to induce DSBs on either side. Afterwards, the unwanted gene simply floats away and the ends are pieced together through non-homologous end joining. Inserting a new gene requires a different DNA repair process, called homology-directed repair, which is only active during the part of mitosis where the chromosome has been replicated, but the two copies have not yet separated. During this time, breaks in one DNA strand can be repaired, without the introduction of indels, by using the other strand as a reference. This allows for the high-fidelity copying of genes to each daughter cell. To make use of this, CRISPR is paired with a donor DNA template, which has the new gene between nucleotide sequences that match the two sides of the DBS. A dividing cell will treat this donor template as if it were the other DNA strand, and repair the break with the new gene inserted. The 4th type of DNA edit, swapping out an unwanted gene with a new one, simply involves combining the last 2 editing techniques. Thus, the CRISPR-Cas9 system gives us basic gene editing abilities. Part 2. Base editing. The spell checker tool. As useful as the CRISPR-Cas9 system is, it has several limitations. For one thing, the indels that it produces can interfere with the functioning of beneficial genes that have been added or substituted. A second problem is that CRISPR-Cas9 editing methods are ill-suited for cases involving very small nucleotide sequences. This is a big drawback, since many genetic diseases are caused by so-called point mutations, in which only a single base pair is faulty. Base editing builds on CRISPR-Cas9 in ways that get around these problems. It could be called a spell checker tool, since it allows for the alteration of single base pairs without the creation of indels. Base editing utilizes a Cas9 enzyme which has been modified in 2 specific ways. First, it has had one of its two cutting domains removed so that it'll sever one DNA strand, but not the other. This new type of Cas9 is referred to as Nic-Cas9 or N-Cas9, since it merely nicks the DNA strand instead of cutting it. Second, the N-Cas9 has had two other enzymes chemically fused to it. These molecular attachments augment the N-Cas9's abilities. One of those enzymes is called a deaminase. Deaminases have the ability to chemically alter amino compounds such as the four nucleotide bases: adenine, A, cytosine, C, guanine, G, and thymine, T, that make up DNA. Scientists have developed two different deaminases for changing base pairs. A cytosine deaminase, which can change a CG base pair to a TA base pair, and an adenine deaminase, which can change a AT base pair to a GC base pair. The way in which this is accomplished is rather roundabout, however. When a cytosine deaminase acts on a C nucleotide, it actually turns it into a uracil nucleotide instead of transforming it directly into a thymine or T base. Uracil takes the place of thymine when DNA is transcribed into RNA. It rarely shows up in DNA, and when it does, it is unstable. Cells have natural repair mechanisms that would ordinarily remove the uracil. To prevent this, a second enzyme attachment, called a uracil glycosylase inhibitor, protects the new base. Meanwhile, the N-Cas9's remaining cutting domain nicks the opposite DNA strand. As the cell's repair mechanisms fix the nick, they replace the guanine or G base with an adenine or A base, since A is still complementary with uracil. Now that the two bases are complementary, the repair mechanisms respond to the uracil differently, replacing it with a thymine. This completes the conversion of the CG base pair to a TA base pair. The conversion of an AT base pair to a GC base pair via an adenine deaminase is very similar, wherein the A nucleotide is first turned into an inosine base. Using the sgRNA to home in on a particular nucleotide sequence, the N-Cas9 can very precisely target a specific base pair adjacent to that sequence for change. By correcting these tiny mistakes in the DNA code, scientists can have a huge impact on the lives of people suffering from diseases caused by point mutations, in many cases outright curing them. Part 3. Prime Editing. The Find and Replace tool. The next addition to the toolkit after base editing was prime editing. Prime editing is the find and replace gene editing tool, as it can be used to find virtually any sequence of about 20 base pairs long and replace it with any other sequence of comparable length. Like base editing, prime editing avoids the creation of indels by using N-Cas9 enzymes to change one DNA strand at a time. Because of this, it can also be used to insert or replace genes without requiring the cell to be in mitosis, allowing for greater flexibility than traditional CRISPR-Cas9 methods. Here, however, only one attachment is added: a reverse transcriptase. Reverse transcriptases are enzymes ordinarily used by retroviruses to convert their RNA genome first into single-strand DNA and then into double-strand DNA before it is inserted into the host's genome. This reverse transcriptase has been modified, however, to omit the second step. Also, instead of a viral genome, the RNA template is supplied by an additional strand, fused to the sgRNA via another palindromic loop. This extended form of sgRNA is called Prime Editor Guide RNA, or PEG-RNA. Prime editing begins with the DNA strand again being opened up so that the sgRNA is lined up with the target nucleotide sequence. The opposite strand is then nicked, creating two flaps. The end of the PEG-RNA contains a nucleotide sequence called a primer binding sequence, or PBS, which anneals to the end of one of the flaps. The reverse transcriptase uses the PBS to recognize its target and responds by transcribing the RNA sequence immediately following it into single-strand DNA, which is added to the end of the flap. This added sequence will contain two parts: an edited sequence, followed by one that matches the rest of the complementary DNA strand. It is at this point that the nCas9 detaches, and the cell's repair mechanisms take over. Because the two flaps are now virtually identical, they compete over which one will be reunited with the intact strand, meaning that instances in which the edit doesn't take will have to be screened out in post. If the edit does take, the unedited strand will be left dangling and subsequently removed by the cell's repair mechanisms. There is now a mismatch between the two strands. To remedy this, a second nCas9 is sent in to nick the unedited strand. When the repair mechanisms fix this second break, they do so by changing the nucleotides in the unedited strand to match the edited one, completing the process. Like base editing, prime editing can be used to correct point mutations. However, whereas base editing can only be used to make four kinds of base pair edits: CG to TA, GC to AT, AT to GC, and TA to CG. Prime editing can also flip a base pair around in the double helix, meaning that it can be used to change any base pair into any other base pair. It can also be used to replace, insert, or delete sequences that are up to about 20 base pairs long. Prime editing has not completely replaced base editing, however, as it is more prone to certain types of errors. Therefore, the two methods are best thought of as complementing each other, giving us a greater variety of options. Part 4. Paste. The drag-and-drop tool. The latest tool to be added to our toolkit is called Paste. This is a very recent development, having been announced less than 6 months ago at the time of writing. If prime editing is the find and replace gene editing tool, then Paste is the drag-and-drop tool, as it can be used to insert large base pair sequences into a cell's genome. The new technique involves the use of yet another enzyme called an integrase. Integrases are used by retroviruses to integrate their genetic information into the DNA strand of the host cell without causing DSBs. Once the virus's RNA genome is transcribed into double-strand DNA, by its reverse transcriptase, the integrase binds to the two ends of the viral DNA strand, forming a loop. It then carries the loop to a specific base pair sequence within the host's genome, called an attachment site. There, it causes a chemical reaction that splices the loop into the host's DNA. Once this is done, the sequence becomes a permanent part of the cell's genetic code. The potential usefulness of integrases for genetic editing is obvious. Scientists have long sought a way of developing them as a tool for inserting therapeutic genes made up of large numbers of base pairs into patient cells. It is even relatively simple to swap out the viral DNA that is attached to the enzyme with synthesized genetic material. The problem has always been the integrase's targeting system. Unlike Cas9, integrases don't use RNA to recognize where, in the host's DNA strand, they are to operate. Rather, they have simply evolved to work with specific attachment sites, with different integrases having different ones depending on viral species. Because of this, it is very difficult to change a given integrase's attachment site. With the advent of prime editing, however, an elegant solution presented itself. First, use the aforementioned technique to insert the needed attachment site into the desired place in the DNA strand, and then use an integrase to insert the larger DNA payload. This is how the new technique got its name, as PASTE stands for Programmable Addition via Site-Specific Targeting Elements. To date, PASTE has been used to insert payloads consisting of 36,000 base pairs into human and mice cells in laboratory settings. However, it is thought that it can be used to deliver even bigger genetic payloads, up to 50,000 base pairs. Using the 1-2 punch of prime editing, followed by integrase-delivered therapeutic genes, they hope to knock out, heritable conditions that involve large numbers of genetic errors. Among the diseases that they named as potential targets for future efforts were cystic fibrosis, hemophilia, the blood disorder, G6PD deficiency, and Huntington's disease. As such, this new technique represents the fulfillment of the greatest promise of the CRISPR revolution. As Omar Abudaya, one of the senior authors of the PASTE study, put it, "We wanted to work toward what gene therapy was supposed to do at its original inception, which is to replace genes, not just correct individual mutations." Conclusion. That completes our look at the CRISPR Toolkit. The truly remarkable thing about the CRISPR revolution is just how quickly the original CRISPR-Cas9 discovery was iterated upon. It makes one wonder what further innovations lie ahead. The law of accelerating returns would suggest that our ability to manipulate DNA may be about to become much more comprehensive. And as our control over the human genome grows, the number of genetic diseases that we cannot cure will shrink. As I close out the video, I would be remiss if I didn't mention, however briefly, the other side of the issue, which is that the same technology which lets us correct genetic mistakes can also allow us to enhance our bodies and minds beyond nature's limits. This prospect scares a lot of people, as it raises the specter of unforeseen consequences, both for individuals and for society. First, I would like to point out that this is merely the latest incarnation of a dilemma that mankind has faced successfully many times in the past. Ever since Mary Shelley penned her famous novel, there have always been people concerned about science going too far and playing God. The fear is that the increasing control that science grants us over nature, including human nature, combined with our hubris, could eventually lead to disaster. Yet, progress through science is self-evident. Even the poorest among us live like kings compared to our ancestors of even a century ago. This isn't to say that science-based disasters definitely won't happen in the future. I mean only to point out that, all things considered, we have a good track record so far. Second, I am of the opinion that, whatever one's reservations about genetic enhancement, they should not subtract from one's support for the development of gene editing technologies like the ones discussed here. There are hundreds of millions of people all over the world who live with genetically based diseases, and the potential for the alleviation of human suffering is too great to ignore. Instead of halting or slowing down progress, we need to have public discussion and open debate about which forms of genetic editing are acceptable and which are not. That, combined with sensible policies, such as prohibiting the editing of germ cells, to prevent such edits from entering the human gene pool, should suffice. We must proceed with caution. But, nevertheless, we must proceed. Thanks for watching the video. If you got something out of it, then please consider giving it a like and subscribing to the channel. You can also follow me on any of these social media platforms. And, if you really like what I'm doing, you can support me on Patreon. The links are all in the description.